Hello, everybody. Good morning, good afternoon, and good evening. My name is Rachel Lieberman, and I am the Director for Global Marketing of Forensics and Toxicology here at Waters Corporation, and I will be your moderator today. I look forward to hosting you all here for this part 3 of our 2025 mid to late year webinar series. Our first 2 webinars were focused on sample preparation and LC troubleshooting, respectively. So today our topic will be on effective and systematic troubleshooting of forensic and clinical LC-MS applications. So before we get started, I would like to go over a few housekeeping items. Here you see a preview of the On24 console for today's webinar. To the left of the slides, you will find the speaker bio, as well as the resource list with additional information and applications related to this topic. Please use the Q&A box located on the right-hand side of the console to ask any questions throughout the webinar. We will be having a Q&A session concluding the presentation where we will answer any questions that have been asked. Additionally, on the right-hand side, you will find the request to earn a certificate of attendance, as well as the webinar survey. Please feel free to fill out the survey at any point during the presentation. We use this information to make sure that we're providing the most up-to-date and informative webinars in the future, so we would greatly appreciate you to take your time to fill that out. We will also be conducting a poll throughout the presentation to gauge some interest and questions about the topic. So if at any point you're unable to see any of these windows, please refer to the icons at the bottom of the page to expand them. Now I'd like to introduce today's speaker. Martin is our clinical support specialist in the Waters Clinical Business Unit, and he brings deep expertise in LC-MS method development and troubleshooting. He is based in the Netherlands and has been a part of Waters since 2018. He has authored a substantial portion of Waters training content and has supported forensic and clinical labs across Europe with hands-on guidance. Today, he's going to speak to you about the effective and systematic troubleshooting of forensic and clinical LC-MS applications. I'm excited as he walks you through the journey of troubleshooting. I'll hand it over to you.
Thank you very much, Rachel, for your kind introduction. And a warm welcome to everybody joining for this webinar. I'm really pleased to be hosting today's session about LC-MS troubleshooting, specifically aimed at the forensic and clinical applications. As you might know, that troubleshooting LC-MS assays can sometimes be a little bit tricky and feel overwhelming, especially when you are not very sure on where to begin. My goal today is to offer you with a sort of a systematic approach that can help you to make the process of the troubleshooting more effective. Now, I believe that one of the most valuable things you can do when it comes down to troubleshooting is to talk with your colleagues, discuss recent changes that have been made to your system or maybe to the method, because no matter how small, this can help in revealing the root of the issue. Sometimes, for instance, it can be something as simple as maybe someone having flushed the wrong mobile phase over your column, and that could result in a decrease in sensitivity that takes up for a few hours before it is restored. To help explore these topics in more depth, I'm also collaborating a little bit, and I would like you to welcome my co-host Martin to the show, who will be contributing with relevant and also critical questions throughout this presentation. Now, let's take a look at the agenda. With the introduction, I'm going to present you with a few topics that you might typically encounter in your lab. And then we will look at the more structured approach on how we can start dealing with those different issues. Then we'll jump into some basics, so the theory behind of LC and MS. And I am very much aware that time is limited today, but I do want to point out some key aspects of these topics, which do have a large impact on both the sensitivity and the robustness of the method, which I think are two key parameters to keep in mind. Then I would like to guide you through a startup process before you do the routine analysis, because I do believe that let me click to the next slide. Yeah, I do believe that prevention is better than the cure, and with these two topics, I would like to go through the reverse troubleshooting, as I call it. Pretty much meaning if you like don't do this or do it differently, then you might run into this and that. And with that, we are already going to cover like about 20 different troubleshooting issues. Now, from there, we go into some more detailed issues that you could be facing with. And I will present you with a workflow on how, for instance, you can deal with a reduced sensitivity in your application. And then we will conclude this webinar, in which I will also give you some tips on how you can even elevate your troubleshooting skills. Now, let's take a look at some issues that you could be facing in your lab. And yeah, I've got Martin helping me out very nicely. So we are seeing this chromatogram over here, and yeah, the peaks have broadened up quite a bit. And Martin is saying, well, I don't know what's happening. Just make up some new mobile phase, and hopefully this will solve the problem. We will dive into specifically this issue later on. It could also be that because of the high sensitive MS systems, that you are more often seeing peaks in your blank. Well, how do we deal with this? Where is this coming from? And it could just be, Martin, that the answer is there right sitting in your hands. But we will take a look at that later on as well. Maybe you have encountered in the past that within one series of injections, you are seeing that the internal standard peak area is decreasing over time. And it could be that you're like, yeah, I have checked all my MS settings, and the conclusion is, oh, it must be an injector issue. Well, let's dive into this in a bit more detail later on as well. Yeah, Martin is looking a little bit upset. Huh? He is a little bit complaining that the sensitivity of the MS is down again. What is going on? And should we call Waters? Yes or no? That is the $1 million question. I hope I can at least give you some guidelines on how to start troubleshooting this, because it might not even that it has to be the MS. Now, whenever you are starting to troubleshoot issues with your assay, I think it would help for you to think about, okay, where could my problem be related to? Is this related to my MS? Because of course we run like thousands, tens of thousands of samples each year on the system. It will contaminate. It will probably see some decreased performance over time. And yeah, it could just be that some maintenance is required or cleaning. But it could, of course, also be related to your LC, which includes the sample, which includes the mobile phase or the column, and possibly the hardware. And yeah, I may have to apologize a little bit for this, but whenever there is samples involved or a mobile phase, well, there is a user involved as well. And of course, he or she could have made some error somewhere impacting your assay. Next to that, we have a computer standing next to our system in which our methods are loaded, and they contain the tune page method. They contain the MS method with the MRM conditions. They contain the dwell time settings, meaning that they are impacting the amount of data points across the peak. And there we have our LC method as well. And yes, unfortunately, those methods, they have to be set up by someone as well. So I hope I don't blame any one of you too much, but there was a user involvement, of course, as well. Maybe before we continue, it would be interesting to learn what you think where the most issues are related to when it comes to troubleshooting LCMS issues. So here's a poll question, which hopefully you would like to answer. So what do troubleshooting issues mostly relate to? Do you think this is related A, to the LC, B, to the method, or C, is it related to the MS? Feel free to start submitting your answers. I will give it some time. So whenever you encounter an issue, is this related to the LC, to the method, or to the MS? I see we are at about 100 answers, but still counting very quickly. So I'm gonna give it like 5 more seconds. So we are at 160 answers. Okay, well, let's take a look what you came up with. So this is actually not that bad at all. So about 40% says problem is related to my LC, samples, mobile phase, whatsoever. 50% says it's about the method, and only 10% thinks this is about the MS. I think that's not a bad conclusion, because this is what I believe is how this relates. But of course, if the method is not 100%, let's say maybe the sample prep is not as perfect, it could result into issues on the LC side, and then at the very end also related to issues with the MS. So they are, of course, also interacting with each other. Well, thanks very much for that. So now let's jump into some basics of LC and MS, a little bit thinking of sensitivity and also robustness of the method. Let's start looking at the chromatogram that we are seeing over here, coming from a drugs of abuse method. We've got like 7 compounds in the mix, and we are also seeing how the gradient is running, expressed as percentage organic. Well, from this page, we can also see that there are pretty much 4 stages as part of this chromatographic method. And let's go through all of these stages in a little bit more detail. Well, at the very beginning, we have what we call the void of the method. So this is where the region where all the unretained compounds are eluting. Typically, this is also the stage where the mobile phase flow is diverted to the waste. Now, it's up to all you method developers out there to make a method in such a way that there is a decent retention for the first eluting compound. And how much that retention is, you cannot just tell by looking at the retention time, but it's very important to calculate what the void time of the method is. And that we can calculate if we know what column dimension we are using and what flow rate that we are applying. So in this example, we are working with a 10 centimeter UPLC column with a flow rate of 0.5 ml per minute. Under these conditions, the void time of the method is at 0.5 minutes. And we see codeine, which is our first eluting compound, it is eluting at 1.5 minutes. And that is a good retention. We also say that the retention factor of codeine is 2. And that's an ideal retention factor for your first eluting compound. That gives enough room for polar matrix compounds to elute in the void, and also to make the the divert valve work properly. Okay, yeah. Martin, so you want fast run times. And what happens if I do not have sufficient retention? Well, if everything is eluting too close to the void, we are going to see a lot of matrix effects because you will end up with co-elution between the polar matrix compounds and the early eluters, resulting in ion suppression, poor sensitivity. It will also result in an unpredictable and an unstable internal standard response. And if the internal standard is not going to be fully able to compensate for that matrix effect, it could also result in poor recoveries. On top of that, when we are not able to correctly set the divert valve to let all the polar matrix to be sent to the waste, we will get an MS, a contaminated MS a lot quicker, leading to an overall decreased performance for all our assays. On top of that, we would need to clean our sample cone a lot more often. So you can tell from this that this stage of the method is extremely important. Then it's time to start looking at the elution of the analytes. And where many people think that this is about separating the analytes between each other, I believe it's much more important to achieve a separation between your analyte compounds and the matrix. There's still matrix eluting across the gradient as well. And the more of an overlap there is between your analytes and the matrix, the more ion suppression you can expect. Of course, the matrix, because we are working in a highly specific MRM on our mass spec, the matrix is typically not visible, but it does elute off the column. And when there's a co-elution, it will give you a decrease in sensitivity. So please don't make your gradients too sharp. Then we have typically also a rinsing step. So after the elution of cocaine, in this example, we are ramping up the flow to 100% B in order to also wash off the non-polar matrix. Think of phospholipids, for instance, when you are working with plasma samples. You want to rinse them off, and also, if it's possible, send the flow again back to the waste. So all these phospholipids do not contaminate your MS. Yeah, Martin, so yeah, this seems like a waste of time, but I would really not recommend to start skipping this phase because of multiple reasons, actually. If you do not use this phase or apply this phase in your methods, you will get an accumulation of matrix on the column, which will result in a decrease in sensitivity. And you might also see a drifting decrease in internal standard peak areas. By the way, if you are working with blood-related samples, whole blood, plasma, whatsoever, it is recommended to work with methanol as the organic mobile phase rather than acetonitrile, because phospholipids, they are much better soluble in the methanol compared to the acetonitrile. So you are much better able to wash off the matrix. Now, the last stage of each LC gradient method is an equilibration phase to prepare the column for the next injection. Depending on the column dimension, the flow rate, this is going to be a longer or a shorter period of time. Also depending on if you are working with a binary solvent manager or a quaternary solvent manager. Binary solvent managers are the faster pumping systems, so they typically require a little bit of a shorter equilibration time at the end. Okay, now let's dive into very quickly into some aspects of MS. So Martin, I don't hope that this is your weight on the scale, 303. Oh, hang on. I see this, that we are looking at cocaine. Right, cocaine. It has a molar mass of 303. And of course, yeah, we are working with the mass spectrometer as well. And it is very important that we tell the software what mass we are looking at in the method. Before we go into a little bit more detail, it's very important to know that for an MS to be able to measure our molecules, we need to create ions out of that. And for a typical basic drug like cocaine, we want to protonate that molecule by adding a hydrogen to the molecule. In this case, so we have cocaine, neutral mass 303. After protonation, this is a mass of 304.4. But this is all about those decimal values, and they are very important. And where are those decimal values coming from? Well, there is a contribution to the decimal value, both based on the isotopes being present in the molecule, but also the mass defect of the different atoms being part of that molecule. Now, in this picture, we can see that the average mass of cocaine after ionization is at 304.4 approximately. But this is the average mass coming from carbon-12 and carbon-13s, both present in this molecule. But our MS is typically set up by measuring only one of those isotopes. This is what we call the monoisotopic mass. This is just a theoretical value that you can even already enter blindfolded as your parent mass in your MRM method. And this 0.2 value, that is coming from the numbers of hydrogens being present in the molecule. Because one atom of hydrogen has a mass of 1.0078. And the more hydrogens there are in the molecule, the larger that decimal value will be of the monoisotopic mass. And we are doing a fragmentation as well in our tandem quad systems. And that typically results in a smaller fragment ion compared to the parent, meaning also that there will be lower amounts of hydrogens present in your daughter. So typically, the decimal value in the daughter should be at the value of the parent down to the 0.0 value. So what you see over here are the correct M/C, mass to charge values of these cocaine mass transitions. These would be incorrect values. So this mass 304.2 is what we call a positive mass defect. Typically, a molecule cannot change to a negative mass defect upon fragmentation. So the 0.7 value is an incorrect value. When we are fragmenting cocaine, we can never add more hydrogens to our fragment ion, meaning the decimal value in your daughter cannot be higher compared to your parent. So yeah, these decimal values, they are very, very important. So yeah, what happens if we are using the wrong decimal values in the method? Well, for sure, this can result in a lower sensitivity overall for that compound. But it could also be that after periodic maintenance, so your system is cleaned, there's a new mass calibration applied, it could be that you're seeing a lower sensitivity after a PM. And not only that, it could be that this is impacting the one daughter ion more than the other one. It could also result in a different ion ratio after a PM. So these numbers are extremely important. Okay, that's all I wanted to say on the theoretical part of LC and MS. Let me guide you now through what I call an efficient system startup, which will not cost you any, I would say, additional time, but it will avoid you ending up troubleshooting some issues. And let's take a look at what those issues could be. Well, whenever you are starting up a routine analysis, the very first thing I would recommend to do is to put in your column and set the column oven to the correct temperature. Then we'll dive into the software and we browse to a project, we open up our tune file and we set the system in operate mode. And we apply the nitrogen gas flow as well. Then it's time to start priming the mobile phase lines, as well as the solvents coming from your sample manager. Maybe just one recommendation to make here. So if you, what I always do is to start priming the solvents when the old bottles are still on the system. Start priming the lines and then take out the tubing, draw some air and only then put it into your new bottle. This will prevent that you are contaminating your old mobile phase into your new bottle, which is good for many reasons. But typically, this step is one that you can ignore unless you are working with methods that have different source temperatures applied in the method. Keep in mind that when you are changing these source temperatures, it will take quite some time for the entire source block to reach that final temperature. So, but typically, I would recommend to keep all your methods at the same source temperature. So you can just ignore this step whatsoever. If there is different temperatures, you would see if you start injecting too quickly, you will see some drifts in peak areas as well. Okay, so when the lines are primed, then it's time to start equilibrating your column. And how do we do that? Well, that depends on if you did that column installation in the very first step, or if the column was still sitting in your system. But I would say in both cases, we do not want to click this load method icon yet. We don't want to run the initial conditions of the gradient to get the flow going. And there's very good reasons for that. I would recommend to work with a high organic flow because, yeah, Martin, that's a very good question. Because if you would start working at the initial conditions of the gradient, well, always your mobile phase will contain some impurities in there or some contamination. Maybe there is still some contamination sitting in your LC or in the column itself. And as long as we start equilibrating at the initial conditions of the method, everything will stay in its place or it will accumulate at the head of the column. And this will result possibly in a lower sensitivity in your initial injections or even a drift in peak areas. It's, I think, much safer to start with a high organic initially. Well, when you have installed a new column on your system, it's not the best idea to go to either the high aqueous or the high organic part of the gradient, because you want to test your system for a leak. And typically, the leak is present the most when we reach the largest back pressure. And that large back pressure is typically obtained when you are working with water to methanol as a gradient at about the 50-50 composition. So when you work with water to methanol, set the mobile phase composition to 50-50 and test for a leak at that composition. With the water and the acetonitrile, it's going to be at 80 water, 20 acetonitrile, where you have the largest back pressure. So test for a leak first and only then go to high organic. then then you have still some time to conclude your sample prep, to prepare your sample list in the software as well. And only then I would say it's time to click this load method icon. And typically 1 or 2 minutes is sufficient time to equilibrate at those initial conditions before starting your sequence. And how is it best? What would be some good recommendations to include in those very first lines in your sequence? My recommendation is to, in all of your routine analysis, to start working with the no injection as the very first line, meaning that you program your method to just run your mobile phase gradient without injecting anything, just to see if a peak is showing up in one of your MRM traces, because that could indicate that there was some contamination in your mobile phase, a contamination of one of your analytes. Then the next one, a blank injection, meaning if there was a peak showing there, it could mean that there was some contamination introduced during the sample prep, followed by a few system suitability test injections, preferably either a low calibration standard or even a solvent standard. Try to see if that SST injection, you can work with trend plot for that. Try to see how that injection aligns with previous injections in terms of retention time and peak areas and peak widths and ion ratios and stuff like that. And when that is all looking fine, then it's time to start running the entire sequence with the calibrators, a blank, QCs, blank, samples, QCs, and again, blanks. The blanks are a very important parameter of your injection sequence. Why is that? I think I already pretty much explained this already. Keep in mind that trend plot is a fantastic tool in our software to keep track on how your system suitability test injections are performing over time. And the very, very last line in your sequence, I would recommend to create a flushing method in that, meaning just program a one-line gradient method, setting it to 100% organic, and just let it flow for 15 or 30 minutes. So all the matrix is nicely being rinsed off the system. There's a few advantages to that. One of them is that storing your column in high organic makes your column go longer, go a lot longer, so you can do many more injections. And this will prevent running into a poor sensitivity because of a matrix buildup on the column itself. Yeah, I hope this approach makes a little bit of sense to you. Now let's take a look at some typical LCMS issues that you might encounter in your lab. One of them is peak shape related. And in the previous webinar series on LC troubleshooting, my colleague Jonathan, he touched upon many of those typical aspects that could have an impact on your peak shape. But one of those is, I would say, even more related to MS. Hang on. Martin, what's going on? Oh, what's going on, Martin? What's there behind your back? Oh, what's going on over there? Ah, I see you're pretending to be a chromatographical peak and you are suffering a little bit from an interference. Yeah, that is a very, I would say, typical issue that we can encounter with LCMS analysis. And this is the chromatogram that I showed you in the very beginning of the presentation. So in this case, metanephrine is showing like a broader peak than what we would be expecting. But what can we do in our method to spot these interferences? Because we do not want to integrate too large of a peak area and overestimate our reported results. So what can we do? And I believe, Martin, you are asking the perfect question. What is my iron ratio? Because it is the iron ratio that will typically indicate if there is an interference present for your analyte. In order to be able to measure an iron ratio, we do need to acquire at least 2 different MRM transitions for the one compound. And then typically we use one of those MRMs for the full quantitation. We call that a quantifier. And the other one we use as a qualifier. And then we just look at the ratio between those 2 peaks. And no matter what the concentration is, it should be a rock stable value. Unless there is this interference present. And then, yeah, that should typically reveal a deviating iron ratio. Because your matrix will probably not show the same fragmentation ratio compared to your analyte. Yeah, so review the iron ratio, but also take care of the smoothing in your processing method. A lot of customers tend to be a big fan of smoothing the data, but I think there is some danger in over smoothing because you are more or less gluing your matrix and analyte speak together. So try to be a little bit gentle with the smoothing and keep in mind that the only reason why you would want to smooth your data is because you would like your automated processing method to work nicely. That is, I would say, the only reason to smooth LCMS-based data. But please don't overdo it because you might miss a possible interference in your data. So in this case, we are looking at unsmoothed data, and I think the interference is showing up much nicely. Also, Martin is now a lot more happy with the interference nicely separated. So yeah, how can we deal with this? So what we saw in the previous slide is that the internal standard, that was actually looking very nice. So it's always good to also compare the peak shape of your analyte with the peak shape of your internal standard. But then if we do observe it, how do we deal with this? Well, like I said, be careful with your integrations because yeah, you might report a too high of a value. What you could do is to prepare your sample again, because it could be that you incidentally contaminated this one sample. If this is happening too often, it might be a good idea to take a look back at your method, maybe change your method a little bit, creating some additional separation between this interfering compound and your analyte, or maybe even look at other fragmentation ions. In this case, we solved the problem by changing the MRM parent to daughter transition, because that resulted in just the interference disappearing. The following topic is about loss in sensitivity. And whenever you are seeing a loss in sensitivity, I think that asking these questions will help you in coming up with an answer quicker. So ask yourself, is this a sudden drop in sensitivity, or was this already decreasing for a long time? And are we now running into issues? Is this affecting only one analyte in the application, or is the sensitivity down for all of my analytes? And is this a problem related to this one assay, or are all my methods performing worse? And how is the background looking? And how is that compared to other series? And is there a signal drift, like a peak area drift, observed within one series? I think if you are looking at those questions, this will help you pinpointing what the issue is a lot quicker. In this case, we have an example of, on the right-hand side, an internal standard with a very much elevated baseline. And that for sure will result in a lower background, even though even the peak height is larger on the right-hand side compared to the left-hand side. In terms of signal to noise, the sensitivity is down. So yeah, what happened to the baseline? Well, in many cases, when you observe an elevated baseline, this is related to some type of contamination present in your system, being it either in the mobile phase or within the column or in the LC system. In this specific case, there was a contamination introduced in the mobile phase by the pipetting tips to transfer the formic acid into the aqueous bottle. Now, if you are very confident that the mobile phase is not the issue, what you could do is to create the magic mix. For those who don't know that, that's 25% water, 25% methanol, 25% acetonitrile, 25% isopropanol, including 0.1% formic acid. And you can rinse your system, including the column, maybe overnight at the low flow rate with the flow diverted to waste, very important. And that will thoroughly clean both your LC and the column. Try to do some injections the next day and see if this indeed has lowered the background. It could also be that when you did a tuning experiment that you are seeing this elevated baseline. But doing a tuning because you expose your divert valve and capillary to quite large concentrations, but that is a typical phenomenon that you can expect. So all you can do pretty much is just to flush the system at a little bit of high organic to get that contamination out of the system. So there's also another example, and this is about a drift in sensitivity within one series. And we have two examples over here. On the left-hand side, we can see the immunosuppressants in whole blood. On the right-hand side, a drugs of abuse method in urine. And what we are seeing is that a calibration curve was injected at the beginning of the series and at the end of the series. And in both cases, the sensitivity of the internal standard is lower at the very end of the series. So yeah, two similar cases, but two different issues that were going on. Now, let's take a look at how to start troubleshooting those type of issues. Well, typically the first suspect is the matrix. So if you are seeing a drop in sensitivity or a decreased sensitivity within one series, my recommendation is to flush the column for about 30 minutes with 100% methanol, just to clean out the matrix, and then perform like 10 or 20 replicate injections of a solvent standard if you have it available, or a low injection volume of a high calibration standard. Then check if your peak areas are going up again, because if they do, it was probably related to an accumulating matrix on the column. And that was the case in the example of the drugs of abuse in urine. So we solved this issue by extending or by including a high organic stage in the gradient and the addition of this flush method at the very end of the sequence. After we made those changes, this method was running a lot more sensitive and robust as well. But there could be something else going on, and that is an effect that can be observed in the MS what we call a charging effect. Charging, yeah, what is that? Well, typically charging is a phenomenon that is observed after your MS is being contaminated. In this case, we can see what we call an ion burn on one of the quadrupoles, and that ion burn will pretty much act as a magnet to for all the ions passing by. And this will typically present itself as a decrease in sensitivity during one series. Now, how can we check the system for charging? Well, first of all, like I said, clean your system with high organic and then perform these replicate injections. But if you then see that there is a decrease in sensitivity again, what you could do, if let's say you are working in positive ionization, then set the system to negative ionization for about half a minute, and then again switch back to positive ionization and repeat those 10 injections. If you then see that the sensitivity is back up again and it starts to decrease again, then for sure your system is showing charging effects. And then it's time to call waters, indeed, to have your system cleaned. This is what was the case for the immunosuppressants in whole blood, and that was solved after a cleaning intervention by waters. So, yeah, time to jump into the conclusion. I hope you find this illustrative for what you were facing as well in your own lab. So my recommendation is to, when you start troubleshooting, is to try to identify to what part of your system the issue is related to. Is it the LC, is it the method, or is it the MS? And when it comes down to troubleshooting sensitivity issues, keep in mind to first flush your column at high organic for about half an hour and then do some repeat injections of a solvent standard. And it would be so much valuable if you have a sort of a trend plot available to take a look at how is this sensitivity lining up with my previous injections. I am also mentioning the ion energies in this slide. They are a part of your tune file settings, but they are also very important when it comes down to the sensitivity levels of the method. So make sure you have set the right ion energy settings. And keep an eye on what the baseline level of your separate MRMs are. Now, if you want to improve your troubleshooting skills, I think it will help to get a good understanding of what chromatography is all about in terms of retention factors, selectivity, efficiency. When it comes down to the MS, try to get a good understanding of what isotopes are, what is a monoisotopic mass, what is a mass defect, and how should I work out these decimal values. Studying the back pressure profiles will help you a lot in troubleshooting retention time issues in your method. Yeah, unsmooth your data. That is something I very often do to check if there is an interference present, if there is sufficient data points across the peak. But also an understanding of calibration curves and peak shape will for sure help you in a better troubleshooting skills. Now, before we jump into the questions section, there is a QR code in this slide, which if you click on that, so maybe you can make a screen dump of this view. This will bring you to a Kahoot quiz in which you can just see with some test cases, some troubleshooting cases, how you are handling with those questions. I think it's a very nice way to troubleshoot a little bit more, and an extension to this webinar as well. Oh, I have this one slide as well. I already forgot. So if you're like, oh, I would like, or I need to learn more about specific topics, well, Waters does have quite a large portfolio of training possibilities available. Either the in-house trainings, as we call them, so provided at a waters facility. It can be on theory, on method development, on troubleshooting. We can also come to your site for a training tailored to your needs. And on top of that, we have an e-learning platform available as well, the Waters Academy Clinical. In the bottom right corner, you can see an example of the same gradient applied on a QSM system versus a BSM system, and why you can expect peaks to elute later when you have a quaternary solver manager compared to a binary solver manager. 50 minutes. There is some time remaining for questions. I hope you enjoyed the content. Rachel, do we have any questions coming in?
Oh, yes, we've had lots of questions. So thank you so much for your presentation. No, it's been fantastic. And our contact information is listed down here if you guys want to follow up later. There are one thing I do just want to mention, because it kind of popped up, is that we do have 2 other like webinars that I mentioned that happened before, and we can provide those links to you if you want to go find them on demand to watch them later. They are on our videos.waters.com platform, but we can also provide that for you. So one of the things that did pop up, one of the first questions kind of after you mentioned it, is if you can restate what that magic mix was for cleaning. Wow. I think you said it fast enough that people were not able to maybe write it down. And so I think people are just interested in what that was to potentially help with that.
This, I believe, is really a Waters terminology. So but this is a mixture of 25% water, 25% methanol, 25% acetonitrile, 25% isopropanol, with the inclusion of 0.1% formic acid. It's a mix of a wide variety of different polarities in the one solvent, and this has proven to be a very powerful rinsing solution for both the LC as well as the column itself. So you can flush your column with that, but please, I should say this again, please divert your flow to the waste, because the last thing you want to do is to flush your LC and column clean and get everything into your MS.
Yeah, no, absolutely. Very, very good. So one of the first questions that we had come in, so this is slightly different, but it's about an LC isocratic method. And they just are curious if the analyte shows up prior to 1 minute, would they still potentially have any matrix effects in an isocratic run?
Well, it's very difficult to tell if you say that the retention time is just below 1 minute, how much retention you actually get out of the void. So I really need actually a little bit more information coming from this. On the other hand, it could still be that if you have quite a bit of retention, so it could even still be that when your peak is eluting at 5 minutes in your isocratic method, it could still result in some matrix effects. It's all about, do you have selectivity between your analyte and your matrix? And if you do experience that, depending on where your compound is eluting, you might want to change the percentage organic in your isocratic method, or you might want to look for another selectivity by trying another different pH or switch from methanol to acetonitrile. All those things that you can modify the selectivity with.
Okay.
But I think maybe to add to that, if you want to check for the amount of ion suppression, I think it's a good idea to inject the solvent standard at one level, and at the same concentration level, inject the matrix standard as well, and then just compare the peak areas. And that should give you an idea on how much matrix effects you are seeing.
Okay. No, I think that's great. And I think kind of ties a little bit into this because you were talking about kind of changing mobile phase concentrations and things. So, and this can, I think in older textbooks or certain things, there's a question that said that never to run a column with, let's say, 100% water or 100% organic, excuse me. And they're just asking if that's still true. I know usually with aqueous, you kind of want to at least have a little bit of organic in there, but I wanted to see if you, had any comments about the extremes per se when it comes to mobile phase.
Yeah. Well, I'm assuming that we are talking about the reversed phase columns in this example.
Yeah.
And it is, well, most reversed phase columns are not capable of working with 100% aqueous. Because it could result when the flow is stopped, sitting in 100% aqueous, a de-wetting process starts to happen within the column with the pores collapsing, and you will lose your retentivity on the column. But there are for sure columns that are capable of working with 100% aqueous. For instance, our HSST3 column can perfectly well deal with 100% aqueous, but our BEH columns, the BHC18 column, cannot deal with 100% aqueous. Now, on the high organic side, well, our columns are even shipped in pure acetonitrile. So I would be surprised that a column would not be capable of being stored in high organic. I even recommend to store it at 100% methanol or acetonitrile, depending which organic solvent you are using as part of your gradient methods. Just store it in the 100% organic.
Okay, that sounds great. So we have a couple questions, and there are a lot that are still coming in, and we really appreciate it. We might not be able to get to all of them, which is totally fine. So we will be able to answer them offline for you guys. But one of the ones, there's a couple that have come in that are related to the internal standard area. And so this particular individual has asked, so how can I solve an internal standard area fluctuation in which the first 15 samples give 2000, and then one sample, it's like, let's say 10, and then it resumes back to normal after re-injection, where it gives a perfect area. Does that make sense?
Yes, it does. Okay. It does maybe seem to me that there was some matrix still remaining on the column that with the next injection, just for the one occasion, is giving this ion suppression effects, which with the next injection, well, was not being observed. So if you do see this more often in your assay, I would recommend to maybe increase the high organic part of the method by a little bit to be better able to rinse off all of the matrix before you do the next injection.
Okay.
I think that's probably where this is related to.
Okay, great. Thank you. Okay, so I'm just trying to figure out this other one. Okay, so there's two. So what is the best way for verification? Oh, excuse me. Sorry, reading the wrong one. How do you choose a tolerance level for the ion ratio? And then on top of that, it's a criteria that most labs have to fulfill, but sometimes some samples come in as saturated. Is there a way that you can deal with the saturation without having to do an additional dilution injection to be able to determine the ion ratio, or do you have to do that?
Okay, good questions. So what tolerance is acceptable? That is, I would say, mostly related to the signal intensities between your quantifier and qualifier. So if you have a very sensitive qualifier, meaning the ion ratio will be close to 1, you can set the tolerance quite strict. And quite strict typically means about 20 to 25%. But if you have only one very sensitive fragmentation and the very next one is a very poor sensitive one, maybe even a 10-fold lower in concentration, then an ion ratio tolerance up to 50% is still acceptable. Now, For the other question related to peaks going into a saturation level of the MS, that will for sure have an impact on the ion ratio as well when there is a saturation effect. But if you do see that quite often, you might want to consider to maybe set your parent mass at one of the isotopic values. So let's say your parent is at 451, you might want to acquire a 452, which is the carbon-13, which will decrease the peak areas. It will, of course, also lower your detection limits, but you can do both use the 451 and the 452, the one for the low-end side and the other one for the high-end side of your calibration curves. Or maybe you don't need that very low sensitivity and you can stick with the 452 and you will not go into saturation effects.
Mm-hmm. Okay, awesome. Well, I wish that we could answer a handful more of these questions because there are a lot, and but we are at time for today. And so we will follow up offline to answer the remainder of these questions. A couple of them are just thank yous because they found this presentation to be very informative. So Martin, thank you so much. And also with that, I do want to say if you have not had a chance to fill out the survey, please go ahead and do so. We ask for any kind of feedback for any webinars that you may want to see in the future. If you want to see Martin and Martin and his sidekick come back for another webinar, and you have some topics or ideas, let us know. We're always looking to make sure we're providing the best content for everyone. And you guys will get a follow-up email with all the links that you need, as well as any kind of certificate that you may need with this. So I want to thank you, Martin, for this amazing presentation. Thank you guys all for your interest and interaction and lots of questions. So appreciate it, and hope you guys all have a great rest of your day. Thank you so much.
Thank you very much for your time.